Hi everyone, welcome to week 4 of your journey into the world of the unseen. Big takeaway from this week is that bacteria are people too. Well, they're not exactly people, obviously, but they are living organisms just like people. So, in other words, they do the same living stuff that you and I do. They breathe, they eat, they digest, they poop, and they have babies. I know. Bacteria respire, they ingest, they metabolize, they excrete waste products, and they reproduce, just like humans. Did I say bacteria have babies? That's right, bacteria have bacteria babies. Sounds kind of weird when I put it like that, of course. The point is, in order to do all of that living stuff, bacteria need genes, just like, well, people do. Now, bacteria carry their genes on a single circular chromosome. Humans have 23, two pairs of 23, or 46, if you like that number better. And so this week we take a look at bacterial genetics and we'll examine how bacteria can transfer characteristics, like, for example, antibiotic resistance, onto their offspring. We actually call them progeny. So below the printed announcement, I have provided links to two short videos to help you understand the concepts of week 4. The first explains the three methods of gene transfer between bacteria. This is kind of the review. Some of you will remember this information from, for instance, a general biology class you might have taken in high school. In the second video, you'll get a very simplified overview of the polymerase chain reaction. We call it PCR for short, which is a process that we use in lab to increase the concentration of a particular segment of genes or a particular number of genes. We don't do the whole genome this way, but there might be a particular set of genes, 4 or 5 or 7, 10, whatever genes that we're interested in studying. We need to make many copies of that segment of genetic material in order to do experiments on, in order to study, in order to do cool science stuff like make insulin and vaccines or combat devastating diseases like cancer. And so this week you will learn how bacteria can be transformed using plasmids. You'll learn the primary steps of the PCR, polymerase chain reaction, and how it is used to amplify genes. And you'll learn to at least you'll get the idea of how to interpret a gel electrophoresis. That is the process that we use to actually read the information from the PCR. Housekeeping items for the week. We're working on topic 4, bacterial genetics. We're back to simulations this week. There are 3 of them. And remember that all of these simulation activities are due no later than Sunday. That's July 13. We're into July now, hard to believe. July 13, 11:59 p.m. GCU time. You have a pre-lab questions for Lab 6 due also on Sunday, July 13, 11:59 p.m. You want to make your initial discussion post, and I suggest your substantive peer responses no later than Friday. That's this Friday, July 11:59 p.m. GCU time. I say preferably by Friday. Of course, the deadline for your peer responses is Sunday, but many students like to wait until Sunday to do their peer responses. And some of you have noticed along the way that I've posted in your comments. If you post earlier in the week, then you're able to participate more fully in discussion. You wait until Saturday or Sunday to do all of your posts, then you really can't get any feedback or any addition to your points of discussion from your peers, because usually by the weekend, for people who post earlier in the week, by the weekend they're done with the discussion, and they might check in with the discussion and tie up some loose ends, but they're basically, those students are done with discussion. They're not up to answering questions. So feedback questions, if you respond to someone, a feedback question is a good thing to help continue the discussion. However, if you post feedback questions on Sunday evening, nobody's even going to pay attention to those, right? It doesn't give anybody enough time to respond to those questions. So the point is moot. Don't even do follow-up questions on Saturday and Sunday, because it just doesn't give people enough time to even consider those. But anyway, that's why I say it's better to do your peer responses before Friday as well. But you know, you're still going to get credit if you wait. But I'm just saying, in order to really get into the discussion, you have to do it earlier. Okay, the big thing this week though is your midterm exam. It's already open. Open this morning. Closes Sunday, July 13, 11:59 p.m. It is proctored. No, you cannot use notes. I think that you can have a piece of paper and a pen or a pencil to do some scribbling, but you cannot refer to your textbook, notes. You can't go Googling anything. Okay, it is proctored. So be aware of that. It's worth 150 points. Complete online and is proctored. Okay, so I did post, along with the videos that I posted for this week for your T4 topics, I did post in class resources a short video that I did several years ago, a midterm topics review. Here's the thing. The the exam is set up. The questions that you get on your exam are drawn from a pool of questions, and you only get a fraction of those. So when I did this video review, I chose the what I think are um, the top priority items, topics that I think you should be familiar with going into the exam. That is not all inclusive, and nor can it be, because the exam changes, the midterm exam changes, as does the final. They change with every class because those questions are randomly drawn from a pool of questions. But I hit the big items: microscope and how to use a microscope and determine total magnification, the gram stain, and how to do a streak plate, and your Kirby Bauer, and all those different main things that we've touched on up to this point. The exam is cumulative, so topics 1, 2, and 3 are on the midterm. Topics from this week will be on the final. All right, so that's that. The midterm exam review is posted, but I also posted a link in my printed announcement for your convenience. But in case you miss it, it is in class resources, so check that out. If you have any questions, concerns, I will be looking at and grading your practical exercises from last week. I'll be doing that this week, and updating your grade books. I see there's a bunch of questions, a bunch of messages that I haven't gotten to yet. I will, and if yours is on there, and if you had a problem with your growing your bacteria, whatever, I'll address those as need be. But so don't worry about anything there. I'll get to you as soon as possible. And if you have any immediate concerns or questions, message 804-229-7993. Use your Halo message box or inbox, and certainly you can email me as well. All right, that's all I have. Keep me posted. Let me know what's going on this week and have a great rest of the week and good luck on that.